Everyone. We're now in the second sitting of the SickKids Discovery Dialogues held here live in the Peter Gilgan Centre for Research and Learning in downtown Toronto at the Hospital for Sick Children. And we are so fortunate to have with us Drs. Batsheva and Eitan Kerem. I first met Batsheva and Eitan now 35 years ago. I think it was 1987.
Summer of 87.
When we first met, when all three of us started our respective training periods here at the Hospital for Sick Children in different ways, and we'll come to that throughout the presentation. Relevant to the theme of our Discovery Dialogue series, all of us were involved or got to see the behind the scenes what was going on in cystic fibrosis research in the 1980s, which of course culminated in the discovery of the cystic fibrosis gene here at SickKids in 1989. We've all heard a lot about this landmark discovery from the likes of our own Lapchi Choi, who led the team, Joanna Romans, who's an emeritus professor here at SickKids in the Research Institute still, Jack Reardon, Manuel Buchwald, of course, who was Chief of Research at SickKids, and many, many others. But I think this will be the first time we hear from behind the scenes what was really happening from Batsheva, who is the first author on the all-important third paper in the Discovery series of papers published in Science in 1989, and Eitan, who was working in the cystic fibrosis clinics here. So this all happened 35 years ago. It'll be interesting to hear their interpretations of the events many years later. So to begin, I want to just first introduce, having Batsheva and Eitan introduce themselves to the audience. And if you can just tell us a little bit about where you're from, really going back to when you were a kid, and then how your career trajectory followed and how you landed here, and then we'll go to Eitan next, and then we'll come back to how you came to SickKids.
Okay, so I'm Batsheva Kerem. Now I'm a professor of genetics in the Hebrew University in Jerusalem, Israel. I was born in Israel in 1955. I studied biology in the Hebrew University and fall in love with genetics. So I did my PhD in genetics on a topic which is very different from what I did in my postdoc. And actually today in my seminar, I will talk about this, which is instability, chromosomal instability and cancer. But when I graduated, I very much liked to go into human genetics. And I looked for a place that will enable me to get trained in molecular biology. It was nothing in Israel in molecular biology, and it just started in the world. And I was looking for a place that can provide molecular human genetics, but also we were looking together for a place that will be good for Eitan, who was already a pediatrician and looked for a place to be trained in pediatric pulmonology. And actually, the only place in the world that seemed to us to be a good place for both of us was Toronto, because other places either had nothing to offer Eitan, or not a good place for Eitan or for me. So we decided to come here. And there is a nice story behind it, because I wrote to several people in the genetics department, which included Ron Wharton, Lapchi Choi, and several others. No emails, regular mail at the 80s. So the first one to answer was Lapchi.
Wow.
And the rest is history. I met with Lapchi. He accepted me as a postdoc. I was quite a good PhD student, so and the rest is history.
So before we go to Eitan, tell us what you've been doing, your current position, and where you've been since you returned.
Yeah, so I spent 3 years in Toronto, and at the summer of 1990, I came back and got a position in the Hebrew University in the genetics department, the same department that I graduated from. I established my own lab, and quite from the beginning, I generated 2 subgroups, one working on CF till today, and the other one, which developed a bit later, on chromosomal instability that I carry on. So I have 2 subgroups. And I carried CF research 30 years already, trying at the beginning to understand which mutations do we have in Israel and in the Jewish and Arab world in general. Then genotype, phenotype, correlation that was interesting at that time. But in the recent years, I became interested in developing therapeutic approaches for CF patients carrying rare mutations that have still an unmet need.
Great. Okay. Eitan, tell us where you come from, and you'd mentioned before the word compromise.
Yeah.
But tell us a little bit about your background.
I'll get into it. So I was born in Jerusalem. I grew up in Jerusalem. I was born in the early 50s, which was a kind of post-Holocaust environment. Jerusalem was a very small city. I loved it. I studied medicine in the Hadassah Hebrew University Medical School in Jerusalem. And then I went to do pediatrics because I love this profession, which is associated with children, innocent children, that you can help them with their future. And we always say that children, at least in Israel, they are 30% of our population, but they are 100% of our future. And then I wanted to do pediatric pulmonology because I thought that something associated with breathing it appeals to me. And we thought, where are we going? Batsheva had the places that she thought are leaders, Stanford, Harvard, the places I thought are good for pediatric pulmonology, had nothing good enough for Batsheva in molecular genetics. So we had to compromise, and we came to Toronto. When we came to Toronto, Lapchi told Batsheva, you came here because your husband has a position in respiratory. Henry Levison, who was the head of chest division at SickKids, told you, you are here because your wife came here to do genetics. Actually, I had no funding at that time. So I received a small grant from the Reichman family, Toronto-based family here, which helped us because we came here with two of our daughters, and helped us to establish our first steps here in Toronto. I think this was the best compromise we ever had in our life.
And just to confirm, you're a medical doctor, an MD, and you're a PhD. And
right.
Tell us, when did you meet in your training?
My best friend's wife was best friend of Batsheva. And when she came to Jerusalem, they told me, look, our friend is coming to study in Jerusalem. Please take care of her. They told me so many good things about her. So the first day that she came to Jerusalem, same evening, I was there asking, who is Batsheva around? And that's it.
Okay, so this is great.
By the way, so far we have 3 daughters and 10 grandchildren.
Yes, and they're all here with you on this trip. And I remember the third was born 1990, was it?
Yeah, yes. Just a few weeks before we left.
Yeah, I think Batsheva was pregnant and she left on a Friday and came back with a baby on Monday, I think.
Yeah.
Okay, so come back to Lapchi Choi was, of course, the lead of the cystic fibrosis team here. He responded to your letter, handwritten letter, and I know why, because I remember your first paper. I read it as a student. It was entitled In Situ Nick Translation Distinguishes to an Active and Inactive X Chromosome, published in Nature in 1983, and your second paper was in Cell, and I think you had several papers, so that's why he responded to your letter, and of course, SickKids has been much to the better because of that. So let's go back to that time. We met in 1987. It was deep. The gene had been mapped to chromosome 7. You came really in the hunt for the gene. Take us back to the time and your interpretation of why it happened here at SickKids. What was the special sauce? What happened here, and what was your role also?
Yeah. So I think that the secret is the teamwork. I think that Lapchi built a team that was very close to each other. And Joanna Romans, who was a postdoc, I was a postdoc as well. She was before me. She knew much more about CF before I came. She was involved in the linkage, etc. And we became good friends and good collaborators. And together with a bunch of technicians and other people, we developed trust in each other. We collaborated, we cooperated, and I think this was our strength. And of course, Lapchi had wonderful ideas, and I think that I brought a genetics perspective. So one of the strengths was to use haplotypes, the genetic background of patients, and this was part of what I was responsible for, and I had very nice discussions with Lapchi, and we built the haplotypes. I just saw the science cover with those colored haplotypes. And I think that this is how we made it.
It was very competitive time for those. There were major groups, many names you would still recognize working on the project. And it was a small laboratory, a group of about a half dozen. Of course, there were collaborators. Manuel Buchwald and Jack Reardon here were involved, but the genetics work was done in a small lab on the 11th floor. And if you saw it now, even our youngest scientists have bigger labs than this.
Yeah.
It was very crowded, and everybody knew. Tell us about those Friday afternoon meetings. You remember where all the data was shown? And
Yeah.
May I add something?
Yes, please.
The few days before we came over here, we hear in the radio in Israel, scientists in London found the CF gene.
Yes.
And we look at each other and we say, Batsheva says, what are we going to do?
Yeah.
And when we came over here, the first thing she asked Lapchi, what's going on? And he said, don't worry, we will find the gene.
So there was a paper published in 1986, I think it was in Nature by a group in London that found a gene, but it turned out that it was the wrong one. And I always say the same story. Lapchi's brilliance was he was the one who saw through that data and knew it was time to keep going, and it was not the right one. And everyone else pretty much stopped. So that's the brilliance of, I think, him seeing what the next steps were and going ahead and jumping ahead at that point.
And persistence.
Okay, well, let's go to you. So you were in the clinic at the time. I remember you used to come up and you'd visit the lab every couple of days, and you brought an energy to the group. And tell us what was happening in the clinic before the discovery and what happened, the day that you heard about it. Was there enthusiasm? We'll come back to the importance of diagnostics and then the discovery of therapeutics, but what happened on that day when you heard from Batsheva that the gene had been found?
I think before the gene was found, there was not much communication between the genetic team that was searching for the CF gene and the CF clinic. Mm-hmm. And Batsheva one day comes home saying, we think we found the CF gene, but something doesn't work. Because when we go over the patients, there is one family that doesn't fit our data. And I asked her, which family is it? She said, family 54. I said, wait a minute, we are not sure they have CF. And that's made the final proof that this was the CF gene.
Because we excluded family 54, and then all the haplotypes, all the inheritance, everything fall in place. So that's an example of our communication at home. And
when the gene was found and announced, there was lots of enthusiasm and hope. And I remember we were sitting at the clinic watching TV, and one of the commentators was asked, what does it mean? And he said, well, within 5 years, there will be treatment for CF. And Henry Levison was the head of the CF clinic, said, he doesn't know what he talks about. It will take at least 10 years until we have a treatment.
Right. Yeah. And how long did it take?
It started about 30 years to have gene-associated treatment, but we don't have final treatment. Yeah. We don't have cure for the patient.
The first drug, Vertex drug for G551D is 2012. So it's 25 years later. Yeah.
So the scientific paper was published in 1989, and as was done for muscular dystrophy with Ron Wharton and Peter Ray's group, even before the paper's group in muscular dystrophy, their research in 85, 86, 87, the diagnostic probes are released to the community and actually used in molecular diagnostic labs around the world. Yeah. So this is open science in a way back 30 years ago and sharing. So the world was following this. You remember when CBC was coming to the lab and filming all the time. The Prime Minister Mulroney's wife was the chair of the Cystic Fibrosis Foundation. Yeah. So everybody's eyes was really on this, and you know, we knew this for a while before the paper came out, and when it came out, you're right, Eitan. There, you know, I think in the community that it was gene discovered, the importance in diagnostics is key, but in fact, a lot of people thought it meant there's a drug, there's medicine, and that took at least 20 or so years. But
I still remember when the SickKids celebrated in the amphitheater in the old building, the cloning of the gene. And in the audience were people from SickKids, but mainly families and patients. Yeah. And when we came, Joanna and Lapchi and I and the rest of the team, they stood and clapped hands. And I was like having tears, and I thought that this is a moment that I will not forget forever. So all the hard work that I did, all the devotion, all the involvement was for that moment that people had hope that with the cloning of the gene, they may have some better future. It meant a lot for me.
But I think there is a very nice story, which is combining this location of the Research Institute here, the heart of Toronto. And maybe Batsheva, tell the story when you were sitting in the pizza waiting for the films to be developed.
Yeah, it was Friday evening. We thought that we had a gene, and we tested the F508del mutation on all the families, excluding family 54. And it was radioactive, and I put it in the film, and I had to wait another hour or so. So I went to a pizza across the building, and I was sitting behind, and I was very excited. What will we find now? Will it be a mutation found in more patients, etc.? And I thought to myself, nobody knows.
People are walking around.
Nobody knows what I feel, what I look forward to. And then I came back to the lab. I opened it. I developed the film, and I saw that 70% of the patients and the families have the mutation. I called Lapchi, and he came to the lab, and
So that's kind of discrepancy. People walking around. They don't know what excellent things and discoveries are being done here in this building.
That's the beauty of discovery, knowing something that others don't. The foundation of discovery. Can you imagine what Darwin must have felt for decades? He knew much different philosophy. But this is incredible. Come back to this 70% of the patients having one mutation. I think this was a real trick. People didn't believe it, right? So it was then ruling out this family that didn't make sense. But ultimately, you found the mutations in the other 30% or so of families. Can you just quickly comment on that?
Yeah. So the first we found that 70% of the patients have this one mutation, but it was important to show that it's not a polymorphism carried by healthy individuals. And when we published the paper, still, Lapchi, Joanna, and I were afraid that after the publication of the paper, someone will come to say, I'm homozygote for F508del, but I am not a patient. And what we can see on the science cover is that we found additional mutations on different backgrounds. So the genetics gave another level of proof that it may be the mutation. All patients with the F508 share the same haplotype, so it's a founder effect, etc.
Incredible. So then you had your third child, Gil, I remember, here, and then you went back home after 3 years or whatever it was, and you had very successful careers. Can you just touch on that and maybe link back to where you're at now? You mentioned you started a company.
Yeah.
And what happened in between in 5 or 10 minutes or less? 30 years?
Yeah. So I established my own group, and as I said before, I had two subgroups working on chromosomal instability and on CF, and because we are discussing the CF, a few years ago, I realized that what we have in the lab may be a potential therapeutic approach for patients, CF patients, carrying rare mutations, because along the years, a company named Vertex, that I'm sure that everyone knows, developed drugs for F508del patients and several others, but there are about 15% of the patients with an unmet need. So we are using antisense oligonucleotide either to skip over an unnecessary exon or skipping on an exon carries a mutation and generating a protein with partial function, but much better than with the mutation. And I realized that if I want it to be translated to a drug, I have to establish a company. I really did not want to do this. I'm an academic person. And actually, Eitan convinced me that I should do this. And with the help of the Hebrew University, I established a company which is called SpliceSense, making sense of splicing using antisense oligonucleotides. And at the moment, we have two projects in CF on rare mutations, and one of them is really very, the effect is very high in tissue culture. We correct the CFTR function and generate a normal protein. Let's see what we get in patients. And
great. Do you want to comment on the last 30 years for you?
Yeah, well, when we came back, I think we first looked to find, identify, and characterize the mutations among the population in Israel. So we have a map of the CFTR mutations among Jews and also among Muslims. And when I was working here, again, Henry Levison, who didn't know how to turn on a computer, but he had amazing database, which was very strong and helped a lot to identify and understand the clinical course of CF. So I thought we need to have also database in Israel, but not only in Israel. So I was elected to be a member of the European CF Society board, and I helped to establish a European database. Now, this was a challenge because you need to take England, who has already database, France, who has already database, and Germany, and get them to collaborate.
Right.
But so we have now European CF database. I established a CF clinic at Hadassah University Hospital. Later on, I became the chairman of pediatrics. Right. And continue to work with Batsheva. So people ask me, oh, how can you do everything, genetics and clinic? And I say, no, we are two.
Oh, that's great. That brings me to my next question is, MD and PhD, husband and wife, how did you make it work? You gave some good examples. Your kids going into science, how did all that work? I think a lot of people are, many of our hires now and the trainees are coming there. Their partners who are also in similar situations. It's tougher now these days. There's so many different options and things. Do you have any advice for the next generation?
Yeah. So it's a challenge to be a scientist, a devoted scientist, a competitive scientist, and also to be a mother. Okay. And without the team at home, we wouldn't be able to do this. Eitan and I shared the raising of our three daughters, and without it, I would not be able to do it by myself if Eitan would not be part of it. We have three daughters. No one went into science. One of them went into medicine. So Gil, that was born here, she is now starting her psychiatry residency. Just graduated from medical school, and our two others are psychologists.
Okay.
So they are all in the same arena, not science, but.
I think it's a kind of no-win situation, being both in academic or clinical or being a parent and scientist, you need to know that you will feel guilty whatever you do. If you are too much in science, you have guilt feelings because of your children or family. And if you are with your family, you have guilt feeling, I'm not doing enough in science. And I need to quote Batsheva. She was asked by a young scientist, if you go backwards, would you do the same thing? She said, yes, I would do the same things, but without the guilt feelings.
If I could.
Well, so, but in a way, you started new projects. I know you're collaborating with and stay in touch with people at SickKids. Do you want to comment on that?
Yeah, so I have kept communication with several scientists, but I built new relationships, especially in the cancer and instability domains. So with Ryan, that I will meet this afternoon, we tried to apply for a grant. We did not get it, but we may now with the progress get it. And with David Malkin, who is working on Lee-Fraumeni syndrome, and we are working as well. So we started a collaboration already a few months ago, and this morning we continued to discuss it. So yes, I highly appreciate the SickKids, the Institute, the level of science here, and I'm very happy to keep in touch and work together with scientists from the Institute.
Do you stay in touch with the clinical group?
Yeah, well, SickKids is a really very special place to work and to gain knowledge. I think as an Israeli, we should be thankful to SickKids because many of the leading Israeli pediatricians had their training here at SickKids. And even now, there are several Israeli physicians. We just got back one of our fellows last week, came back from 3 years here at SickKids. And soon I'm going to meet Felix Rachen, who's the head of the chest department here. And we will continue collaborating, doing research together, and also sending our young doctors to be trained here.
Yeah. So for those of you who are viewing, we hear a lot about at SickKids this concept of precision child health, and really the model for me is the cystic fibrosis discovery. It's to find the genetic underpinnings of why a disease or disorder comes about, use that in diagnostics, and then use the information to develop new therapeutics. And in fact, cystic fibrosis is really the model. It's the first one that went from start to finish. There's examples in cancer too, but this was the paradigm everyone followed. And of course, Francis Collins in the United States and others were involved in this. And in a way, it set the foundation for many, many other gene discoveries. Even the Human Genome Project really was that. So, this happened at SickKids. So I want to hear a little bit more, like dive deep into the why. You said some good examples. It was the team, but for me, it comes back to the people, and you were one of the people. I was an observer, kind of watching. I was kind of the coach, on the sidelines, giving advice and things. But what do you think back on? What do you tell your students?
I would tell my students that if you have a dream, if you have an aim, don't be afraid to pursue it. It may be challenging. There were days that I thought we won't find the gene. And keep going. If you believe in what you do, if you love what you do, if you are fascinated about science, go for it. And I unfortunately see among my students, and many of them are female, that are the best students, but from family reasons, or they will not pursue their dream and they will not continue with science. And I think that what I learned here is to be devoted, persistent, dedicated, and keep your good work.
Well, that's very nicely put. And Eitan, so again, from my perspective, when the discovery was made, I really didn't think there would be a drug that would come because the biology was so complex. But it happened. And you want to, what did you think? Did you think there would actually be a medicine that would come out of this at some point?
Well, I'm a believer. And, this drug by Vertex, we had a patient that was on the transplant list. She is a mother of 2 children with oxygen. She started this drug. We got it as a compassionate drug for her. And within 2 months, she was off oxygen, starting exercising, and later on had another baby. So we can see it as a miracle. And I think you should believe that you will find it. It will come at the end. And just to add to what Batsheva said, I learned a lot from my patients. And one of my patients had lots of difficulties, which I'll not get into, but I told him, you know, I admire you. You had so many difficulties and you made it. And he said, no, I do not have difficulties. I have only challenges. And it's a state of mind. And if you go into science or medicine and you look forward and you think I have a challenge, I will overcome it because it's a challenge. It's not a difficult, it's a challenge. So this is another sentence that if you want it, you will find a way. And if you don't really want it, you will find many excuses.
I don't think I could say that any better. That was just perfect. Were there any questions that have been emailed in? No. Okay, so we're good. Do you have any closing comments? What do you Batsheva is giving a scientific talk in about a half an hour here on her work on fragile sites, and that goes back to your PhD in a way.
Yeah.
So what comes around.
Right.
And any other, what really excites you now? What are you going to do the last leg of your career?
Yeah.
Or the next leg of your career?
Yeah. So I'm very much into the chromosomal instability and cancer, trying to unreveal the molecular basis of instability and I got just the NIH grant for this, so I'm very excited about it. And the second arm is developing therapeutics for CF. If we will be successful, it will be a whole circle for me, closing a circle, but it's too early. So that's what I see in the next 5 years or so. And I really thankful for the good training, education, mentoring that I got in the SickKids, which helped me to become a mature scientist.
For me, continuing taking care of patients, doing research in order to understand better the disease, which hopefully eventually will develop into drug for them, is something that I always did and I will continue doing.
All right, maybe we'll just close now. Do you have any final, you shared some of your special stories of your time in Toronto, anything else you remember? I remember one, but I'll leave it. You go first.
Well, I think as a family, we had fantastic time in Toronto. As a physician, I had amazing experience. Somebody very smart told me before I came here, you should be like a sponge. Just absorb, absorb, absorb. And when you come back to Israel, just put it back and get everything out.
Yeah.
Yeah, that's perfect. I remember, so coming back to the concept of the team, I had just started, I didn't know anyone in Toronto. And the very first weekend, do you remember when we met in Hyde Park?
Yeah.
Do you remember that?
Yeah,
I was riding through Hyde Park and I just met you.
Yeah.
And you were there with your family, and it was the first connection to real people I had here other than the university. And it came back to, I really agree that it was the special people involved in the team, and it went into the clinical side too, and you were the connection. I remember when you came up, everybody was happy to see you. And we didn't talk about the 200 the families used for linkage, but that was all collected on the clinical side and provided to the laboratory group. And Mary Jury and others didn't get a lot of the credit, but all that had to be in place. And it was the team right across the entire hospital, not just in the institute, but right across the whole hospital. And it happened when both of you arrived at SickKids. So all the credit to you too. Let's close there. Thank you so much for joining us, taking time out of your vacation, and happy birthday to you.
Thank you.
Okay, thanks everyone for joining us, and we'll see you next time.
Thank you.